This information is going to cover the Microscopes and Cells lab. You can find this information in Chapter 4 or some correlating information. Make sure that you take these notes before we conduct the Microscopes and Cells lab. Please bring your handwritten notes to class when we are going to conduct the Microscopes and Cells lab. This is the first lab that we're going to find within the Bio Lab Introductions Part 2. You can find the Bio 111 Lab Introductions handout on D2L if you look under the Content tab. Let's go ahead and start discussing microscopes and cells. And we're going to add some information about a cell and then draw a very basic picture of a cell. All living things are made of cells. This is called the cell theory. There are lots of different types of cells, different sizes and different shapes. Different sizes, shapes, and functions. But while there are lots of different types of cells, all cells will have a few things in common. The first thing that all cells have, no matter how different they are, is going to be something called a cellular or plasma membrane. The second structure that we are going to find in all different types of cells will be some type of genetic material. Genetic material refers to molecules like DNA, and RNA. Most cells are going to have the genetic material called DNA. There are a few different types of cells that will have a genetic material called RNA, but that is going to be less common in cells. The third structure that we see in all different types of cells will be a structure called ribosomes. Let's make a note that viruses do not contain ribosomes. In fact, if you review Chapter 1, you're going to find that viruses do not have a lot of characteristics of life seen in other cells. Viruses do not have ribosomes, and all cells have ribosomes. Viruses hijack cells and use the host cell's ribosomes. Viruses do not meet a lot of the characteristics of life, and you can review Chapter 1 to determine that. Let's now draw a picture of a cell and discuss a few features of the cell. I'm going to make a big circle. And this is going to be my cell. I am definitely not an artist, so do the best you can, and I will too. The outside of the cell, or should I say the lining of the cell, is called the plasma membrane. You may also call it the cell membrane. I'm going to abbreviate that PM for plasma membrane. The plasma membrane forms a lining that separates the inside of the cell from the outside of the cell. This is important because we want to ultimately control all the activities inside of the cell so we can maintain stability, or what we call homeostasis, inside of the cell. Let's list the function of the plasma membrane of the cell. We're going to describe the plasma membrane as separating the inside of the cell from the outside of the cell in order to maintain homeostasis of the cell. Homeostasis is a term that was introduced in Chapter 1. Homeostasis refers to maintaining stability within the cell to keep the cell alive. The inside of the cell is what we call intracellular. That term intra means inside. The outside of the cell is what we call extracellular. And extra means outside. Therefore, the plasma membrane forms a lining that separates the intracellular environment from the extracellular environment. All cells have DNA inside of the cell. And we will discuss the location of the DNA inside of different cells shortly. Remember that DNA and RNA are what we call genetic material. This genetic material was inherited after some sort of cellular reproduction activity. So oftentimes DNA is called the molecule of inheritance. Molecule of inheritance. So now we've talked about two things that we see inside of the cells: plasma membrane, genetic material, and we only need to draw one more thing inside of the cell, and we're going to draw ribosomes. Ribosomes appear as little dots inside the cell. The dots may be individually found inside of the cell, or the dots may be found in chains. The dots may also be found bound to other structures inside the cell. These little dots are called ribosomes. And they are very important to the cell because these are going to be the sites for protein synthesis. Human beings have at least 23,000 different proteins that they need in order to function normally. This means that synthesizing or assembling proteins is very important to a cell. The ribosome is the location at which protein synthesis occurs. Let's continue on to question number 2 and discuss 2 major different types of cells. We have a lot of different types of cells, but all of these different types of cells that we find globally can be characterized into one of 2 major groups of cells. These 2 major groups of cells are called prokaryotic and eukaryotic. I'm going to rewrite the terms next to each other and draw a prokaryotic cell next to a eukaryotic cell. We are going to make a drawing of a prokaryotic cell and a eukaryotic cell so that we can compare similarities and differences. I'm now drawing the cells. One difference that you see right away between the prokaryotic and eukaryotic cells would be that prokaryotic cells are much smaller. And eukaryotic cells are much larger. That is going to be one difference that distinguishes prokaryotic from eukaryotic cells. Let's continue to compare the 2 different types of cells. Both cells have a plasma membrane. So that means that they have some similarities. Remember from the last problem, we said that all cells have DNA. Well, the prokaryotic cell has a clump of DNA that is coiled and condensed, and it's going to be called a nucleoid. The eukaryotic cell has DNA that's going to be found inside of a structure called a nucleus. So for the eukaryotic cell, the DNA is found inside of the nucleus. Let's go ahead and write that down. That is a notable difference between the cells. Prokaryotic cells have no nucleus. Instead, their DNA is coiled and condensed into what we call a nucleoid. Whereas eukaryotic cells have a nucleus with DNA inside of it. The environment inside both cells is not dry. It is usually an ooey gooey fluid type of environment. This is going to be called cytoplasm, the semi-fluid that's found inside of a cell. Just think of it as an ooey gooey type of fluid that has a lot of molecules and maybe some structures inside of it. So both cells have cytoplasm. The prokaryotic cell have additional pieces of DNA that appear as small rings. These are going to be called plasmids. The bacterial cells, which is a type of cell that falls under the group of prokaryotic cells, contain plasmids. A plasmid is a specialized ring of DNA that codes for specialized traits. Plasmids code for traits that are not necessary for the functioning of the cell. Plasmids contain DNA which ultimately may code for specialized traits seen in bacterial cells, such as antibiotic resistance. A major benefit of plasmids for humans would be that plasmids can be manipulated or genetically engineered for human benefit. We will discuss this later in the semester. Eukaryotic cells do not usually have plasmids. Let's continue to make some drawings in our eukaryotic cell. It is a very large cell and it has a lot of structures inside of the cell. All of these weird shaped structures that I just drew inside of the eukaryotic cell are going to be called membrane-bound organelles. Organelles. Prokaryotic cells are so small that they do not contain the organelles. So that's going to be another difference between the prokaryotic and eukaryotic type of cell. I will go ahead and draw some dots indicating ribosomes inside of the cell, inside of both cells, since both cells have ribosomes. If you look at both types of cells, based on the notes that we have written so far, hopefully you will understand that the eukaryotic cell has a lot more structures inside of it. This is going to end up making the eukaryotic cell more complex. The last thing that we want to do is discuss which type of species of organisms have prokaryotic cells and which species have eukaryotic cells. So which groups of living organisms are made of prokaryotic cells? This is going to be bacteria and another type of cell called archaean types of cells. Bacteria and archaean organisms are going to be composed of prokaryotic cells. Please read a little bit more about archaean types of cells in your online textbook. In this class, we will briefly discuss bacterial cells in order to compare them to plant and animal cells. The types of organisms that are made of a eukaryotic type of cells will be plants, animals, fungus, and another type of cell that we call protist. Protist type of cells are basically the type of cells that you can find in watery environments like ponds or streams. Amoeba, paramecium are example of protist type of cells. There are many more examples of protist type of cells. In this class, we're going to mostly talk about plant cells, animal cells, and bacterial cells. Our emphasis will be on plant and animal cells, but we will mention bacterial cells every now and again in order to compare. Make sure that you bring these handwritten notes to class when we complete the Microscopes and Cells lab.